The aim of this procedure is to be able to determine the proportion of cells in the different phases of the cell cycle. This is accomplished by first adding the cell proliferation labeling reagent BRDU to a viable cell culture for incorporation into newly synthesized DNA. Next, stain the BRDU in the cells that have incorporated the reagent. Then, the DNA is stained with propidium iodide. The final step is to evaluate the proportion of cells in the different phases of the cell cycle by flow cytometry. Ultimately, the cell cycle distribution of a population of experimental cells can be visualized through flow cytometric analysis of BRDU-labeled cells.
The main advantage of this technique over existing methods, like tritiated thymidine incorporation and scintillation counting, is that with BRDU labeling, the proportion of cells in each cell cycle phase can be determined.
Visual demonstration of this method is critical, as the flow cytometric analysis steps are difficult to explain without accompanying images of the different parameters that need to be adjusted.
Demonstrating the procedure will be two graduate students from my laboratory, Cecchini and Mehdi Amiri.
Add BRDU at a 1:1000 dilution to the experimental cell cultures. Incubate for 1 hour. To harvest the cells, aspirate the culture medium and then wash the cells thoroughly with PBS 2 times to remove all traces of medium. This time using PBS supplemented with 3 millimolar EDTA, rinse the cultures quickly once more and then aspirate the PBS EDTA solution thoroughly. To detach the cells, add around 0.5 milliliters of PBS EDTA to each 6 cm culture dish and incubate the dishes at 22 degrees Celsius